Okay, good morning to all of you. So, I think we are starting for the division Cyanophyta. Uh, in relation to the Cyanophyta, here you are observing the Cyanophyta, particularly cyan means the blue, and formerly it is to be known as the blue-green algae. As these are to be firstly treated as the bacteria because they are mostly closely related with that of the prokaryotic bacteria than that of the eukaryotic algae. As if we are discussing for its evolution, that is the history, so it is to be supposed as about 3.5 billion years old, that is having 1 billion is equal to 10 years. They are to be dominated as biogenic reefs. A reef, that is the it is to be like that of the which growing on the these rocks are there. Their age of the bacteria, it is supposed as 2.5 billion years to 750 million years, right? So they are the widely spread. As the multicellularity, it is generally the characteristics, and therefore the cyanobacteria were the first algae. As due to their microscopic characteristics, they are commonly found in the marine sediments. Then pelagic zone, that is the open sea or the ocean part is there. It is called as the pelagic. Commonly also found in the fresh water lakes, and they are also in the live in the extreme environments, also in the chemically and by the temperature. In distribution, some cyanophyceae members like the pools, ponds, Anabaena, Rivularia, already they are generally seen. Then secondly, the distribution endophytic, then endozoic, cryptophytes, epilithic, sapropelagic, and generally in the loose lying agaropilus forms are there, or the ball, and the lichens are there. I think we have seen in detail. So remember these examples for the as a cyanophyce. Then importance for this one. So there are the two photosystems and to produce the organic material and give the oxygen as a byproduct. And very important that the evolution of the Earth's oxidizing atmosphere. So I think they play the important role in the evolution of the planet. Then in importance mainly atmospheric nitrogen into the usable form. So it is mainly done by this cyanophyceae members are there, and because of the atmospheric nitrogen is unavailable to the living organisms, because breaking the triple bond is difficult. So according to Gudoco phenomena, so it is defined as the variation of the colors in some cyanobacteria with reference to change in the wavelength of light. That is, as falling of light's different wavelengths, the cyanobacteria appears of the different colors. For a good example is the oscillatoria. In characteristics, chlorophyll A, phycobilioproteins, phycoerythrin, phycocyanin causes the, or it shows the blue coloration. And secondly, there is having the allophycocyanin. The storage generally having of the glycogen, cyanophycin granules are there, and cell wall is made up of amino acids and the sugars. The different forms are there: unicellular, colonial, filamentous, or multiseriate, right? So, and secondly, trichomes commonly due to their mucilaginous sheath and due to the cells are the isodiametric. Different features are there. The mucilaginous sheath, it functions protect the cells from the drying and the involved in the gliding, right? And the sheath is often shows the different color: red color due to acidic condition and the blue color due to the basic, whereas yellow shows the brown color with the high salt content is there. So just see the how the coloration of the these thalli are there. In the range of vegetative structure, here you are observing the different photographs. So non-filamentous single cell or palmelloid colonies, unicellular spherical colonies, mucilage is the of different size. So you see anacystis, it is magnified and natural. Then Crucoccus, it is showing the unicellular, but the colonial form is there. Then Synechococcus, all these are unicellular forms. Then unicellular Gleocapsa, you see the natural photograph and the magnified portion. Then there is a Gleotheca is there. Colosphaerium is there, right? So natural and this one. Then Aphanocapsa, palmelloid stage. Palmelloid, that is, it forms a irregular colony formation. It is called as a palmelloid. Microcystis, it is unicellular. Eucapsis, it is a 3D diagram. So it is for the eucapsis. In trichomes, oscillatoria is the good example. See the natural one and the magnified line sketch is there. The Lingbaia is the another good example. The empty cell, empty sheath is present and the trichome is there. So with reference to spirulina, commonly we are calling as a single cell protein. It is detected, but in morphology it is a trichome showing the ribbon shaped chloroplast is there. Then Arthrospira, it is also the same, it's showing the irregular zigzag type. Okay. Then Rivularia bulata, which is commonly seen on the rock, is there. See the diagram of the Rivularia. Then Gliotrachea. Gliotrachea we have seen as it is single, it is with heterocyst and this one. So you see the how the position of the heterocyst is there or how the other cells are there. Alright. The other normal vegetative cells. Then microcoleus and the cyanophycean nostoc in order. So you see this is a nostoc and it showing the specific type of the trichome. Then same member from the nostocales is the Anabaena. Anabaena, it is the one of it shows heterocyst, slime mold, decaying cell and vegetative cell. Cytonema shows the pseudo branching due to the sheath, mucilage sheath is there. You see this is a magnified diagram of the cytonema. Then there is the branched pseudo branching is there, right? So branching in cyanobacteria, it shows the specific type. False branching is there, and commonly it having the outgrowth of the filament adjacent to the dead or the specialized cells are there. The true branching outgrows from the cells that changes their axis and division of 90 degrees from the axis of trichome. The cyanobacteria, cyanophyceae members, Stygio nematales. So you remember the diagrams properly and natural position. Then there are the habit of the coastal ponds are there. So it shows the Apellosyphon species. Then another one is the Mastigocladus. So it shows the true branching. It is reported in someone. Then in vegetative and asexual reproduction. So vegetative reproduction mainly seen by means of the cell division or fission. Then fragmentation, that is the trichomes get into pieces, fragments, polycystis, crococcus and the microcystis. Then vegetative mode of reproduction, cell division or fission by means of the there is a separation of the cells are there. The filament or the colony breakup into the small fragments are there. The vegetative mode of reproduction, hormones are there. There is a space in between two cells, or they shows there is a specifically is developed into the double concave disc between the two adjoining vegetative cells. So it causes the hormones. You see that this magnified photograph of the oscillatoria. So any vacant cell, you see that this is the oscillatory filament with hormogonia. Vegetative mode of reproduction, it is the important. Then pseudo hormogonia is are there in the some Vestella intricata species. It shows the such type. Then endospores, that is the asexual reproduction by endospore. So why asexual? Because the water conditions, pH, temperature, salt concentration, it is going to change, and therefore it effect on the normal gametes development. So there are the asexual spores develop. This is the Dermocarpa. It shows the asexual spores. And then there is a chemosiphon is there. Then those spores are there. So you see the position of chemosiphon and their character. Then in sexual reproduction, akinete formation. What is akinete? So it is a thick-walled resting spore, function resistant to unfavorable environmental conditions. So this is a magnified part is there. Then in spirulina, what is spirulina? It is a commonly nowadays used in the single cell protein, and it shows a major for the flamingo population, and commercial as a food source is there. The cytokinase in cyanobacteria, neurocytokinase block the neuron transmission in the muscles. The good examples are the anaviana and the oscillatoria. And heterotoxins are there. They inhibit the protein phosphate, cause the liver bleeding, and find in the drinking water. So this is our today's lecture. In the next lecture we will see about the heterocyst. Okay. So thanks to all.